Hey guys, hello and a very warm welcome back to my channel, Avan Anjo. So I'm back with another video. So in this video we'll be discussing another topic of UPSC Zoology optional. So all of the people who are new to this channel, a very warm welcome to you to my channel. So I have also begun a series of UPSC Zoology preparation for the optional. So all the biology background people who wanted to take up this topic in their mains, so they can subscribe to this channel. I'm coming up with all the videos and I would be completing all the topics of zoology optional within a month, within 2 months most probably. Okay, so also I've created many videos of general studies already, like polity, constitution, international relation, okay, internal security, so you can check them all out. So in today's video I am going to discuss about the topic that is Golgi apparatus. I've already created the videos starting from the basic, and this is basically going in a series. In the previous lecture I talked about endoplasmic reticulum, okay, where we have discussed everything relating to it, its structure, its function. And video which was even more previous to that video, we have discussed about the topics of what are cells, prokaryotic cell, eukaryotic cell, nucleus, biomembrane. So you can check all those topics out, okay. And this is like a series, we are going on in a series. So now let us begin with this topic. So Golgi apparatus. So I've created some images, okay, so that you guys can understand it. Here you can see this interconnected cisternae like thing, which is endoplasmic reticulum, and they are rough in nature, okay. Let us consider they are, this one is the rough endoplasmic reticulum where the ribosomes are lying above it, okay. Now this is the rough endoplasmic reticulum. Now if I am going to compare it to the Golgi apparatus, you can see that there are similarities between endoplasmic reticulum and Golgi apparatus, and it is said that the Golgi has emerged from endoplasmic reticulum, okay. Now if you guys can focus on this, so this whole is the Golgi, and when you see in the nucleus, like if here it is the nucleus, so Golgi are found lying next to the endoplasmic reticulum. So in the previous video we discussed that these ribosomes that are there on the endoplasmic reticulum, they are going to do the protein synthesis part, okay. So when they are going to do the protein synthesis part, so they are going to synthesize proteins and they are going to lie inside the lumen of the endoplasmic reticulum. They are going to undergo certain modifications, and then they are going to be passed on to the Golgi apparatus. Now how are they transported, how are they passed on? I told in the previous video also, it happens through vesicles, okay. So there are like vesicles get pinched off from the endoplasmic reticulum, they get pinched off, okay, and they start to move towards Golgi, okay. Now these vesicles, I think I should focus it more. Yeah. So these vesicles, when they are moving towards the Golgi, okay, so they are moving towards the Golgi. These would be carrying like the protein of interest, okay. Now they're going to move towards the Golgi. Now what would happen is that these vesicles are going to get fused with the Golgi, okay. So okay, so if I have to draw it, so you could say that it is going to go and get fused with the Golgi, okay. So it is going to attach to the Golgi, okay. Now when they get attached to the Golgi, they are going to release the protein that is inside it, okay. Now this protein would be getting released, and in the same way the pinching off is going to happen. Here the modifications of the proteins are happening, the packaging of the proteins are happening, the sorting of proteins are happening. I'm going to talk about the functions differently. Right now I'm just going to show you the pathway, how it goes, okay. So it gets vesicles get attached. Now the phase which is receiving, the phase that is the phase of the Golgi that is receiving the incoming proteins from the endoplasmic reticulum is called as the cis phase, cis, okay. Whereas the phase which is going to liberate the protein from itself is going to be called as the trans phase, okay. So you need to remember this, cis and trans. Now this phase is also called as the, like you could say, it is like a phase that is in a way it is going to receive, okay, and this phase is going to liberate it. So this phase is hence also known as maturing phase. Okay, so now let us come back to this. So the vesicles gets attached in the same way. They are going to get pinched off, they are going to go to the other one, they're going to get pinched off, going to go other one. So you can see that these ends of the Golgi are swollen as compared to this, which are quite tapering. Okay, so this tells you that the pinching is going to happen, it is going to move to the next one, the pinching is going to happen, it is going to move towards the next one. Okay, so this is how this happens, this is how the vesicles move. And while they are moving, so they are going to carry certain proteins, okay, like they would be having certain localizing proteins. Okay, there are going to be certain localizing sequence. Okay, so these localizing sequence, what are they going to do is that they are going to be leading to they are going to be actually be a kind of a label. Okay, they are going to be a kind of a label, like when you go into a post office. Okay, so they are going to you are going to add a label on it, which is going to consist of that address where you need to send that post. Okay, like that label only, this is the label. Okay, this localizing protein sequence is going to be the label, and this is the label that is going to tell that where the protein has to be delivered. Now this sorting part, okay, this sorting is called as sorting. Now this sorting part is done by the Golgi. Okay, they are going to be acting as the post office, and they are going to deliver the vesicles or the proteins, not the vesicles, basically the vesicles are going to get merged with the body of the Golgi, and the protein is going to be given out. So this work where the mail is going to come, and they're going to the mail is going to come to the post office, that is Golgi, and then it is going to be sent to the respective locations where the address is there. Okay, like according to the address. So this is what is the work of Golgi basically. Now this is how the vesicles goes, and this is how the transportation works. Fine. Now if it is clear, so let me just talk a bit about the structure parts. Of course, this swollen part is where the pinching off of the vesicle is going to happen. These all are the cisternae. Okay, and it is acting as, you know, there are certain things that are happening in the Golgi, like chemical modifications. Okay, chemical modifications are happening. Also there are going to be the sorting of proteins, okay, that are going to be happening. There are going to be packaging. Okay, so packaging and then sorting. So all of these stuff is happening inside the Golgi. Fine. So Golgi, out of the Golgi, what is going to be emerging is going to be the lysozyme, which I'm going to talk about in the next video. So lysozyme emerges from Golgi, and they are also the vesicle like structure which consist of acidic hydrolytic enzymes. So I'm going to discuss about that also. They are called as the suicide house of the cell, where the cells are going to be digested. So these are like the digestive organs of the, they are going to digest all the debris and all the things that are not required in the cell. They are going to digest the cell also, and they also can, they under they are autophagocyte also. So they are going to, you know, eat themselves up also after their work is done. So we are going to talk about that in another video, that's for another video. In this video I hope that this much has been clear to you, how the Golgi works, what is its work, why it is there in the cell. So you should put all of these explanation into better words when you are writing, of course, but this is the basic concept behind the Golgi apparatus. So I hope that this much has been clear to you. In the next video, as I told, I'll be talking about the lysosome. So till that, I hope you all would be staying safe, stay at your homes, and be happy. And also please don't forget to subscribe. That is the most important part actually. Just subscribe right now, go hit that subscribe button, so that I get to know that yes, you guys are liking the videos and you want me to make more of it. So go subscribe, leave a like if you understood it well, and share it with your friends also who need the explanation, who want to know the concepts. Thank you so much guys, have a very nice day, bye.